Is it visible, sir, slides?
Yeah.
Hello?
Yeah, visible.
Yes. Sir, shall I start, sir?
Yeah, sure.
Yes.
Good afternoon, one and all present here. I welcome all of you for a second lecture on successful groundnut breeding. Today we have with us Dr. M.V.C. Gowda, former professor and head Department of Genetics and Plant Breeding, US Dharwad. To begin with Sir's brief biodata, Sir has completed his graduation and post-graduation from US Bangalore and PhD from IARI in the year 1985. And Sir has started his career as a research assistant in AICRP on millets at US Bangalore. And later on, he went on to join as assistant professor in US Dharwad in 1986 and 87. And in 1987 to 90, he worked as a plant breeder in AICRP on oilseeds at US Dharwad. And he went on to become associate professor and finally professor and head in the Department of Genetics and Plant Breeding at US Dharwad. Sir has also guided so many students, like 30 M.V.C students and 20 PhD students. That is too much, to be honest. And Sir has also has nearly 70 research papers in journals of repute to his credit, and so many other publications also. Sir has got so many awards and recognitions right from his undergraduation. He has got a gold medal for his undergraduation from US Bangalore, and a junior research fellowship for post graduation, senior research fellowship for PhD, and Sir is also the fellow of Indian Society of Genetics and Plant Breeding, and he has also awarded with Counselor Award by Crop Improvement Society, Ludhiana. Sir has also got incentive award by US Dharwad for mobilizing the highest funds from the external sources. Sir has also developed so many groundnut varieties, including GPBD4 and Mutant 28-2. Sir has also developed some mapping populations for late leaf spot and also rust. And also he has expertise in mutation breeding as well as marker assisted breeding. Sir has also visited Tuskegee University, USA for molecular markers in groundnut related training program. Sir, welcome you on behalf of DG IIGR Junagadh and AICRP groundnut. Before begin with Sir's lecture, let us, I request Director Sir to give some introductory remarks.
Thank you, Dr. Manjunath. Sir, Dr. Manjunath is a new scientist, fresh scientist in discipline of agricultural economics, and he is also product of US Bangalore or Dharwad?
Dharwad.
Okay. So, sir, this initiative is the first of its kind in IIGR as well as ACRIP groundnut. And specifically, we were discussing several things while interacting ACRIP scientist, ICAR scientist, and IIGR scientist. So there we show that some generation gap, idea gap, generation gap among the newcomers, or they are more technology savvy like computer, phone, all those things. But basic plant breeding or concept or how to utilize in the field, they have a very good knowledge. Particularly classroom knowledge, book knowledge, but how to articulate in the field and get the result or develop varieties or coming to the aspiration of the society, farmers or country, somewhere we are missing that touch. So once we are discussing, then immediately we thought, why not to have a interaction type of talk or lecture between the uh, old, previous experienced scientist, not only breeder, all the discipline, production, protection, and improvement, and how they have developed, breeding the varieties, new lines they have developed, integrate different traits from here to there, or multi-trait parents they have developed, and how those developed varieties rule for 30 years or even 40 years. So we have to have some kind of interaction which are missing with the new generation or new system of functioning. So that was the genesis of this idea, so that we can have a good interaction, because nowadays regular interaction between the former professor, experienced breeder with the new generation may not be feasible physically. So now this corona, after corona period, we are very much versed with this interaction, online meeting, Zoom meeting, like that. So we thought, why not we can request our former professor guides, so they can give some tips, idea to the new generation, so that they become more productive, or they can plan properly, or they can use resources on priority basis, and they are able to deliver what society desire from them, or institute or ICR as a whole required to address the specific problem related to groundnut. So that was the genesis. Then we discussed who are the our um, former workers in the CRIP system who has delivered, contributed a lot and meaningfully also. So you are one of them. Naturally, it come first in our mind that Dr. Gowda will be the our best choice. So we requested all the former groundnut breeder, worker or scientist one by one. Sorry, your lecture will be the second one. First one was from Dr. Nadav, and similarly from Janila and Nigam, Muralidharan, and all those former people. So we one by one we are arranging. And last lecture, last talk, the Dr. Nadav, sir, our Zoom capacity was 100, but more than 100 people tried to join in that meeting, but we failed because our Zoom subscription was only for 100 persons. But now we have increased around 300 or 400, so there won't be any problem. So this is about this program. And regarding Professor Gowda, nothing, no need to new introduction or nothing. I will tell 2 or 3 points to the newcomer. Dr. M.V.C. Gowda, Dr. Bharsne, and Dr. Radhakrishnan, our former director. These are the trio in groundnut breeding who has first started the molecular breeding work in India. And from there, anything, whatever we are seeing here, molecular, marker, or groundnut, these 3 persons are the pioneering pillar in groundnut from 3 different places. One from ICAR-IIGR, one from DGR, IIGR currently, and US Bangalore. Sorry, Dharwad. So they have started, they prepare a good team, good population, and they work together. And first map of that RAST and LLS ICAR-IIGR published, and from there we first got the marker and marker assisted selection in groundnut. After that, GPBD4, that is the first variety having a interspecific background in India, we released. And recently probably all you have seen what is the contribution of that GPBD4. And again US Dharwad, that Dr. Bhatt and his team, that they have published that paper, what is the contribution of that GPBD4 and markers, related marker, associated markers, and QTL. And after that, a huge breeding program, molecular marker assisted breeding program. And from that we again started DGR and US Dharwad and ICAR-IIGR, that hyolic breeding, and lot of work US Dharwad has done, we have done, and ICAR-IIGR, and the outcome is the Gidnar 4, Gidnar 5, then GG40, 39, and so on. So sir, will be happy to listen you, hear you, and more specifically the younger breeder, those who have joined recently or worked 3-4 years experience in groundnut. So they would be happy to have a more and more specific tips for field activities, or how to augment their breeding program, parents, and deliver a good variety which can rule at least 20-25 years in the system. So that is we are missing. And as we all know, the groundnut area in the country is gradually reducing. Earlier, when you worked, sir, that time it was 8 million or so hectare in India. Now we have come down to almost 5 million hectare. But demand of production is more. We have to produce more from that 1/3 or 1/4 area. But we have increased our productivity. We have crossed 2000 kg per hectare average national, but some states we are getting more. So our task is very critical. We have to produce more from the unit area. So we need to have a climate resilient varieties which can contribute or produce more under stressful condition. And this crop, as we all know, this is a upland crop and dryland crop, legume. So always suffer from different biotic and abiotic stresses. So with these words, I once again welcome you, sir, on behalf of my IIGR, myself personally, and entire all the scientists from ACRIP and all ACRIP centers, all the 30 ACRIP centers. Sir, thank you. I welcome again, and you can start now. Floor is yours, sir. Thank you.
Thank you, sir. Thank you so much, sir, for your introductory remarks. And over to you, sir, Dr. M.V.C. Gowda, sir.
Simply, I'm overwhelmed. So nice, Dr. Bera Saab, to invite me to share my experiences with groundnut fraternity, especially as you said, youngsters. I will be too happy to share my experience and straight away. And thank you so much for all kind words that were expressed about me and about my work. And let me start, and there will be sufficient time for interaction. You can continue to interact. Anybody can, they are free to contact and discuss anytime. No problem, no issues at all. Let me share my experience in groundnut breeding. I will go with my slides. Please allow me to share my slides, or yeah, request has been sent. how to share.
Dr. Manjunath, you allow to share?
Yes, sir. Yes, sir. I have already given permission, sir.
Okay. Yeah. Visible?
Yes, it is visible, sir.
Yeah.
You can go ahead.
Yeah. So this I term it as a journey with groundnut breeding. It was a passion for me, of course. And more specifically, of course, different aspects have been tackled, or we work together with so many people. But foliar disease resistance, one area where we spent a lot of time and where impact was also felt. And I will concentrate on breeding for foliar disease resistance. Here, of course, I would like to just, as Bera Saab said, share my experiences with the development of GPBD4, the first foliar disease-resistant Spanish bunch variety uh, that has made some very good impact in different areas, as you said. And the second aspect, of course, for quite a long time, when we started, as he said, there was nothing as far as genomics is concerned, and we started the work, and we had to really generate a lot of tools, starting from the scratch. And my colleagues at US Darwad have taken it forward further. After uh, I went out of university to take a project coordinator job at Millets at Bangalore. But I still continue to get associated this work. That was the passion in me. Let me start. Of course, everyone knows about foliar diseases, and when we talk about foliar diseases, early leaf spot, late leaf spot, and rust. And late leaf spot and rust, these are very widespread, and they cause significant yield loss, uh, affect fod recovery due to damage due to pegs, as well as death of plants. They predispose crop to soil-borne diseases, affect the quality of produce. Obviously, we all know that the best solution comes from resistant varieties. So, in my journey, I call it as my journey with groundnut breeding. It's all started with my doctoral program, PhD research program. My thesis titled Experimental Evaluation of Concepts Related to Breeding for Rust-Resistant Productive Populations in Groundnut. I worked at for my research at IRA Regional Station. Now it is IAMR, Military Research Institute. Uh, my guide was Arunachalam. He was National Fellow working on groundnut. So I acquired very deep knowledge on designing of experiments, data analysis, and interpretation. Very famous quantitative geneticists, biometrical geneticists. And then, of course, there was Dr. P. Subramanian, groundnut pathologist uh, at TICRISAT. And part of my work I did at TICRISAT also. And from him, of course, a lot of good things were acquired. Creation of artificial epiphytotics, creation of differential treatments to assess rust and leaf spot. As I said, they occur together, it becomes very difficult to decipher which is affecting how much, etc., and which is more important, etc. And for this, we went with 4 different treatments: presence of both the diseases, presence of one of these diseases, and absence of both the diseases. This created a really good condition. And then another aspect which I acquired from him, which is very, very important, is disease measurement, both quantitative as well as converting that into scores, which we'll just spend 1 or 2 minutes there in the next slide. So what is the sort of knowledge I gained out of this work? Very experts in different fields, and good conditions were there, good facilities were there at ICRISAT to conduct experiments, and with available material over there, we did some work, and I came to know that foliar disease resistance, when we talk about it, it is mostly quantitative in nature, and obviously, there are different diseases, like in this particular case, rust and lead spot. I always talk about lead spot occur together, and one I got insight into interaction of these diseases, how to decipher them, etc. Then interrelation between disease and yield and its components is very, very important for us as breeders, because unless it has got impact on yield, and even if the variety is not yielding so very well in normal conditions, then that will not be accepted by the farmers. So obviously these aspects are very important, I came to know. Then importance of, that's what I said, importance of stability of yield under different conditions. Then most important, of course, is interdisciplinary work, pathology and breeding. Likewise, many other disciplines as we go on, we will see. This is very important. So this I acquired during that period. So tell a few words about modified disease scale. This most of you who are dealing with these diseases know about this. Pathologists especially know about this. But at that time, very sketchy knowledge was there, and Subramaniam really developed some very innovative ways of scoring diseases, as well as generating data which is quantitative in nature, which could be used, say, for example, in modeling or in deciphering the relationship quantitatively between diseases and yield and its components, etc., is quite possible as compared to, say, scores 1 to 9. So this was the benefit I acquired from that. Scale is, of course, even now being used. One can also generate quantitative data using defoliation, which is very, very critical. We'll come to know. And then one can estimate RGLA, that is remaining green leaf area, estimated on main stem at different points of time. This is also very important to gain deep insight into the relationships. And then mean disease area scored on remaining leaves. This gives us quantitative data. So that could be converted into 1 to 9 scale as depicted in this particular slide. Then in 1986, I moved to US Dharwad, where I spent nearly 23 years. And this was a place where diseases occur naturally. Very ideal spot for really to work on these diseases. This is one thing. The other aspect, of course, I was posted initially in the teaching department, and later I came back to teaching department only, which I will narrate anyway. So Dharwad being a very good spot for a good center for screening or developing something on foliar diseases. So that was added in attraction for me. Then when, of course, apart from teaching, we are expected to do some research and start some research for long run, so that we can work and do some impactful research. So that was the aim of uh, having research along with teaching. Then what I did, of course, since these diseases occur naturally, with my experience, background and experience during my PhD work, I started a small experiment on yield loss on different varieties grown over there. To tell you one another aspect of Dharwad is it comes in what is known as transitional zone between high rainfall area and the dry areas. In between it comes up, and we get well distributed rain, good soil moisture is available, and that is one of the reason also we get a lot of diseases also. So here when I went there, and I saw that double cropping is in practice. Early varieties are grown, not just long duration varieties, mostly Spanish bunch and early varieties. And after harvest, second crop of chickpea or other crops used to be taken. And many a times, say for example, sowing takes place very early in the second week of June, ideally, of course, depending on the rainfall. And crop will be ready by October. Sometimes it gets caught up in rains also. That's also there. That problem I will just briefly touch upon later. So when we conducted this small experiment, we found that all the Spanish bunch varieties which are in vogue there under cultivation, they were all susceptible to both the diseases. And in 2 years, it was quite significant. Yield loss was quite significant. So what we really felt was that tackling this particular problem can lead to very impactful research. This is one thing what one should really decipher in the beginning itself. Then, of course, after 2 years, I was moved to ACRIP as groundnut breeder. This was an excellent opportunity for me because facilities were there, resources were really good, and there was a multidisciplinary team. It continues to be a multidisciplinary team at Dharwad now, even now, important center. And at that time, under University of Agricultural Sciences, Dharwad, there were two centers, major centers, Raichur and Dharwad. Raichur, of course, got separated, but these were the two competing centers also. So it gave me opportunity also to interact with groundnut fraternity research workers at national level for 3 years. Then, more important, of course, linkage with farmers, extension agencies, especially millers and seed producers. This is very, very important. As Berasab said, seed is a really critical issue, which is a big challenge in groundnut. That's the whole problem. And millers play great role in terms of market conditions, as well as, say, our seed system could be there, breeder seed, foundation seed, certified seed, etc., could be there. But millers also provide big chunk of seed directly to the farmers, as well as, of course, farmers themselves. They store a lot of farmers store their own seed for their own crop. So this is the scenarios for a seed is concerned. So it was a good experience for me to learn a lot of these issues and develop contacts also during this period. So what was the status of breeding as far as foliar disease resistance is concerned at that time? So as I said earlier, large area, and in this particular belt, transition belt, Spanish Welsh cultivars were ruling, and all of them they were really susceptible. When we looked at any available resistance varieties, there were some varieties of Valencia type like ICGV 86590, etc. They had rust resistance, but obviously they were not accepted by the farmers and also millers, especially because of uh, undesirable pod and kernel features, and shelling out turn was less. Obviously, market formulae was not ideal for those, especially in those areas, for these Valencia cultures. Some VB cultivars, that is Virginia bunch cultivars, were having resistance, fair amount of resistance to both rust and late leaf spot, but they were all long durated and not useful for that area. That was the target area for us. So strong need was felt for developing Spanish belt cultivars, which are all highly susceptible for resistance to rust and late spot, and they should also possess good agronomic and pod and kernel features. So that was the theme, focus. So what did I do during those 3 years? Of course, as far as other things were also taken up, but as far as foliar disease resistance is concerned, there was a lot of material available, breeding material available, produced, generated by earlier breeders like Abib, etc. And Nadav, my colleague, was also involved. He was working there as a junior breeder when I joined as groundnut breeder. Many crosses were available. There were people who were expertise in skilled in crossing. This is one important issue, of course, always crops up, as we all know. Groundnut breeders producing crosses, it's a challenge again. So there were good skilled people who could generate crosses for their So I started focusing on FDR specifically with my students, like initially students like Abdul Qadir Prabhu and Ramesh Bhatt, they worked on few aspects of already available breeding material. They worked on mating systems, uh, as well as handling segregating generation in early generations especially. What is the response to early generation selection as far as yield and resistance is concerned? These were the focal points. So what we learned from this, plant selection was effective as far as plant habit is concerned, even if you make crosses between botanical types. It is a big challenge again, crosses, transferring traits between botanical types for obvious reason, complexity. And then more important pollen kernel features, these plant selections were effective. Plant selection was also useful as far as resistance is concerned to foliar diseases, but it needed real confirmation through family evaluation as well as appropriate environment. This is very, very critical, crucial. Many places it may not be available. So obviously we can't aim to develop varieties quickly for foliar disease resistance. Then we learned, obvious to everyone who are involved in breeding, not only groundnut or any other crop, family selection and replicated trials for seasons are essential as far as yield is concerned. And some of the yield related, right? Like in groundnut, maybe shelling out turn, etc. Because they're highly influenced by environment and large scale trials should be there to confirm. So, what was the situation scenario? Out of this breeding material, actual selection did not meet our requirements, farmers' requirements. Obviously, there was a deficiency. So, we went for one has to go for. We felt that one need to select right parents and generate large number of crosses and also large segregating populations to begin with. So by this time, of course, I was again went back to teaching department. Maybe people felt that I needed there for teaching, and also, of course, I had a passion for teaching, especially genetics. So I went there, but it gave me an opportunity to focus only on specific issue, that is foliar disease resistance. But there were some issues, of course. Anybody who works in teaching departments, they know that there is always fund crunch. So it became a habit for me to write projects for external funding. And subsequently, at a particular point of time, there should be at least one project to carry on our work. That's what the aim was. And many projects were, of course, funded by ICR, DBT, PRNS, etc. during my tenure. Then, but most wonderful thing, people, students, young students, continuous stream of students year after year, and young students ready to translate our ideas into real work with all enthusiasm and commitment. And it is very easy to influence youngsters, inculcate in their minds and hearts about the importance of this work. So that was the greatest advantage working with in the university and in the teaching department. Young students will come to know their contributions from start to end. So to begin with, in 1991, we assembled germplasm from acrip and icresat, resistant germplasm, evaluated for disease as well as some yield rates, selected four important germplasm at that particular point of time. One landrace, Valencia landrace 259747 from South America. Then other three derivatives of interspecific hybridization, very crucial as far as foliar disease resistance is concerned. So all these CS16 originally developed at North Carolina University by Starker. Later, this material was transferred to ICAR-IIGR. ICAR-IIGR facilitated good distribution of this material. GBFDS 272 again from Microsoft, DG101 from Dachlam, of course. A lot of good initially cytogenetics work was carried out over there, and material was also there. So crosses were affected with three Spanish bunch varieties, GL24, DH40, all were early, KRG1. So here, one interesting thing I would like to say about Ramesh Bhatt, who is so sincere forever, and also his contributions will be known at the end of this, by the time we reach the end, in the sphere of foliar disease resistance in general, as well as genomics of groundnut. So I just asked him, he was working on something else, some segregating material, but I asked him, it would be nice to learn crossing technique, and why don't you produce these crosses, 12 crosses, I said, and as sincere as ever, even today, to the core, he is so sincere, and he produced these crosses. So that's the role of course, youngsters also. And then in the subsequent 2 years, F1s were evaluated, famous line into tester analysis. Extensive data was collected on diseases as well as yield and its component. Analysis was done, some insight was obtained. Large F2 populations in each cross was raised. Individual plants were assessed for habit, disease reaction, pod and kernel features, as I said earlier, at plant level. Plants combining Spanish bunch habit, desirable features were advanced. This work was done by one of my M.V.C students, Kuchunu, who is now a dean in agriculture college. And what we learned here also is that quite good number of segregants which were potential were produced or identified. And our F1 performance parameters were indicative of what we are going to get in F2. That's the interesting thing. So F1 can give insight into what we are going to get in F2. Then continued the work on FDR. Sorry, I have gone back. Okay, advancement of breeding material. So systematic evaluation of selected F3 families was undertaken for yield and its component and disease resistance by one of my students. So family evaluation is really important, and good number of selections were made. Desirable segregants number got reduced. Here on one of my other student who has contributed quite a bit during his MSc as well as PhD, and later he served at various places as groundnut breeder, including CRISPR. So he has done very systematically artificial epiphytotic screening, and then yield potential, generating the data on yield potential and yield loss, etc. Then some 3 superior lines were selected. They were subjected to large-scale trial. And GPPD4, what we are all talking about, was identified at this particular stage, and then pushed that into multi-location trial. In the university, there are many centers where at that time there were Raichur and Dharwad, and material also coming from places like departments. So we pulled together this material and subject that to multi-location trials. And from there, of course, selected material goes to active trials. That's what happened with respect to GPBD4, along with other lines from other centers. So it was conducted for 3 years, 2000 to 2002, and released for cultivation. To tell this story in a short way, GPBD4 was released for southern India, what then it was known as Zone 5, as well as Eastern India, West Bengal and Orissa. And but it became popular in transitional belt of North Karnataka. Obvious reasons will come to know. Though it was identified and released for all of these areas, why it became so popular in transitional belt of North Karnataka? This is again very important issue as far as plant breeders are concerned. We need to really understand the agroecology, where the crop is grown, what are its requirements, why some varieties performed so very well, what are the traits, how we can improve those traits in those varieties. This is the issue what one really look at it to really come up with some impactful research and advancement of research. This is really important. But another other issues, of course, is it has taken long time, 11 years, and you all know that environmental condition, ideal conditions, artificial epiphytic, all these are essential. This is a very tough challenge again. These issues were there. So this particular small paper of 4 pages, three pages, was published in International Research Newsletter, published from ICAR at that time. No longer this journal is there. It was a good forum to gain information. In the sense, not just looking at very huge papers and their contributions, their impact factor is important. What's happening really on ground? This was a very good journal, International Rights Newsletter. So, wherein we were learning a lot about what is the small things that are happening in different places, and how we can imbibe that information, etc. I think it is not there, but it is wise enough now to really come forward and publish such a newsletter so that what's really happening at ground level, not visible in the big journals where everybody looks at, is very important. So this particular paper has been cited so many times. We'll come to know about it also, and quite often this is referred because whenever people talk about GPPD4 or whenever they use this material for some purpose, maybe basic work, maybe for breeding, always they quote this. Whenever people talk about success of interspecific hybridization, starting from Starker or anybody, they quote this because one of the successful stories. So I urge the breeding fraternity to start some uh, newsletter like this for our own benefit. Okay, what made GPPD4 so successful in transitional belt? And as I said, in this transitional belt, moisture is not a big problem many a times, and diseases are very much prevalent, and we need to have varieties which are resistant, and they will have real good impact. That's what happened with respect to PPD4. High yield, resistance to LLS and rust. Apart from that, of course, it matured early, may not be as early as other earliest varieties, but maybe a week more it took, but very high yield. Then other features were high pod growth. You'll come to know in the picture. And partitioning coefficient and shelling out turn, these are very, very important, especially shelling out turn for millers or kernel recovery is a very economically important component. Desirable pod and kernel feature always this plays key role. You all know in groundnut breeding, this has to be kept in mind. A variety may be super variety in terms of yield, but acceptance again, really one of the important aspect is pod and kernel features. So this has to be taken into consideration always. It had 49%, 49 to 50% oil content. Oil recovery was really good. In fact, I know of many cases millers offering 100 rupees more for GPBD4 as compared to other varieties for oil recovery. This is a very important aspect. Oil ratio at that time 1.6 was really good as far as Spanish bunch varieties are concerned. It was around 1 because it was based on this CS16 or ICGB86855 interspecific derivatives. This has many good features of this kind. They also went along with the disease resistance in the making of this particular variety. Then one other interesting feature was very high level of protein, high level of protein and high oil content. These are important features. Of course, economically this could not be felt, but obviously a cake groundnut cake, quality cake one can get, and only dairy farmers can visualize this. Then, more importantly, especially in the areas where these diseases occur, quality of fodder gets affected drastically, and quality of this fodder was very good in this, obviously. So these are some visuals of variety when it's compared with susceptible variety, ideal or desirable pod and kernel features comparable to one of the long appreciated variety TMU2 comparable. Then you can see visualize the pod bearing of this variety. Some features might have come from CS16 also. This is very important. Then, of course, extension agencies have they played a very great role in popularizing this variety. The picture is there. And in some places, it was grown in 150 to 200, even 300 acres farmers' field in this transitional belt and quality fodder. So important issue in this, of course, always we emphasize whenever we talk about this variety is the yield loss as compared to other varieties. And see with respect to all the yield components. But some insight which is really required. Let's get back to the original data. Here we have JL24, TME2, TAG24, ICGV 86590, which is resistant to rust. M28-2 mutant, somebody spoke about it as contribution. It is resistant to leaf spot. GPBD4 resistant to both the diseases. As far as yield under protected condition is concerned, you can see some level of improvement is there in these later cultivars. But yield loss is pretty low in all these, depending on the disease, rust, leaf spot, rust and leaf spot. So there is a gradual, so you can very well imagine the importance of both the diseases, and more so late leaf spot especially. So this is a key issue, and the economic product recovery is very high, or yield is very high, significantly high under disease condition. This is a wonderful thing. Coming down to kernel yield, we have a lesson to learn here. ICGB 86590, it was fairly good when we looked at pod yield, but when we look at kernel yield, it has come down to match the other non-resistant varieties. So obviously there is some problem here. The problem is Chelling outturn and pod and kernel features, obviously. So this is quite important. So at various conditions, varieties should perform very well. And obviously in all this, GPBD scored very high under disease conditions as well as in protected. So we even with respect to oil, I talked about oil percentage. That's very important. GPBD4 was good both in protected as well as unprotected conditions, epiphytic conditions, significantly superior. Further, in yield under protected conditions, it may not be so very good as compared to the other varieties because it has got some high partitioning ability. So obviously, it fodder yield may not be, or it is just touching other varieties. Of course, the list was in TAG 24, highly determinate variety. But when we come down to the fodder yield, in the disease condition, it's significantly superior, and quality, nothing to beat. Polio disease resistant varieties are always good under disease conditions. So this is about it. Then Bera Saab also talked about, as groundnut workers, we all know that seed is an important thing, especially because seed multiplication is very low. Our seed rates are very high. Groundnut and soybean, they suffer from this particular challenge. So we need to have strong seed production chain. And we are fortunate to have very good, a model, so to say. In different circles, they say model seed unit, the Dharwad seed unit, of course, established by S.A. Patil, and his vision for farmer-friendly developments really touched us a lot of benefits in soybean and groundnut, where these problems are there, and see the type of breeder seed production which went on for years. You can see in this. And farmer participatory seed production was an important component here. So that's very important. And of course, I had put this picture earlier, 2004. And in 2024 also, I came to know that a lot of area is there under this particular variety. Seed coming from their own farmers saved seed, as well as millers, apart from reduced, of course, seed chain because of for obvious reasons. There could be better varieties, and new varieties will definitely taken importance. They should be given importance. So under that scenario, even now it persists. Dr. Bera was talking about 20, 30 years of variety. Of course, nothing to beat PME2 and such varieties. They have stayed for since 1940s. So, but in this particular condition, transitional belt, this variety is still popular among farmers. That's the important uh, conclusion that I wanted to draw from this particular. So here I want to just remember other players, especially just recently, one day before the earlier scheduled talk, one of my colleagues, P.D. Shankar, who worked in NDDB, and later he was advisor to Karnataka Seeds Federation, where he was advising on seed production and seed supplies by Karnataka Seeds Federation, both in groundnut and sunflower. And he did human service in popularizing groundnut and also producing uh, huge quantities of foundation seed as well as certified seed. And his linkage with farmers was so good. And that was one of the key reasons for rapid spread of GPBD4 in transitional zone of Karnataka. So I just remembrance for his contributions and memory. This I have put this particular picture, and also it indicates the role of seed production agencies, be it government, public, or even private, like millers, etc. They play a huge role. So obviously, one has to tap those resources and develop good linkages with them. Working together definitely can yield results, quick results. And of course, Bhaira Saab also mentioned about this scientific and economic impacts of GPPD4. This is where we are really missing out, be it national institutes or universities or breeding establishments, whatever it may be. We have failed to really put our success stories more efficiently. So one small attempt has been made by Ramesh Bhatt and his team. Many of them are also workers on worked on groundnut with me, also Matigi Naidu in this picture you have. They have also worked with me for on groundnut and others. So they have, with all constraints on data, etc., they have come up with some very nice article. I want to see more of such articles, and also that too based on actual data, field ground level data, to showcase the efforts of our research fraternity. It is not just breeders alone. So achievements, any achievement in any aspect of groundnut, for example, should be projected, and there should be real effort in this direction. I urge youngsters to really come with such impactful assessments. let me just go into little details about this impact, economic impact. So what they have done really is they've taken breeder seed production as a barometer, a base over years for 17 years from 2006 to 2022. Some 1,654 tons of breeder seed has been produced at the university, converted to foundation and certified seed with multiplication ratio 10. Of course, this is a conservative estimate. As I said earlier, farmers saved seed, especially at the later phases, and millers, they do contribute depending on the demand by the farmers. So farmers' demand is really very important. Impact at farmers' level is really important. So that we need to really take into consideration. As far as this estimate is concerned, of course, the area covered with 40 kg per acre, and then production estimate based on very interesting paper published by KVK Shumagga, who have conducted 60 demonstrations with 60 farmers, 24 hectares over 5 years from 2005 to 2009. And there they have compared GPBD4 and TME2, and the difference in productivity or yield level mean average was 25.94 for GPBD4 quintals per hectare, TME2 21.43, 20.90% superiority. So based on this and using the price prevailing at that particular point of time was taken as a base, and value of TME2, value of GPBD4 for that particular area was calculated, and benefit was also shown, additional benefit accrued 1,752 crores over 17 years. So this is a conservative estimate. So this sort of a picture one should develop. So many successful varieties are there in so many different places. So if we can really collate and come up with such scenarios, it will be wonderful for our fraternity, grounded fraternity, to show others, showcase our successes to others, maybe administrators, maybe extension agencies and administrators especially. So this will go very nicely in the long way. So coming to other impacts, as I said earlier, one of the first varieties to be developed, which was under cultivation, which was used in various basic studies, in genomics and in variety development. Many varieties tracing back to this GPBD4 were developed, 5 varieties there. And then many populations, mapping populations were developed, traits were mapped, different traits, principally deadly spot and rust, and even other traits depending on the other parents were mapped, and many publications came. And you see right now a variety getting 500 citations. There could be more for such varieties, but this is only an example I am showing that visibility could be there and there could be appreciation. So this is what is required. Impact studies are really required. So this finishes the first part. So we'll get into the second part, genomics assisted resistance breeding. So we came to know that prevailing conditions, the environmental conditions, and many years are required to achieve some success, whether there is a way to enhance the efficiency of this, reduce the period, and also enhance the efficiency. Can we undertake quite widespread breeding in different places using these tools to develop or to add polyadicea resistant to other positive features is a question that we tackle. And it's a long journey. Of course, we all know about the benefits of genomics assisted breeding, the problems with FDR, that is foliar disease resistance, as far as conventional breeding is concerned, quantitative nature of resistance, interaction of diseases, significantly influenced by environment. There is obviously always linkage drag. We have to overcome this. And the resistance could be distributed for different diseases, pests, etc., in different germplasm, even if they are there in the same germplasm like GPBD4. But when we trace them, when we track them, they will be independent. Obviously, we need some tool, efficient tool to track them efficiently. Overall, phenotypic selection is less effective. We all know about it. So, genomics assisted breeding can really enhance is the mute point. Of course, you all know about these things as students of modern plant breeding, but we should not forget about the arts of plant breeding. Spending more time with the plants, learning more, you'll come to know spending more time can lead to a lot of nice things in the next few minutes. Um, increase the speed and efficiency, linkage drag problem, pooling or pyramiding resistant genes, recovery of recurrent parent in backcross breeding, wherever we are interested in developing some essentially derived varieties. Then to select QTLs when we are talking about quantitative. So our target is not always genes, it is QTLs, and the markers associated are identified by identified being QTLs. Then, of course, it can be a substitute, especially in the early phase of breeding for phenotyping, even in the absence of say diseases. We can really boil down to few potential plants or lines which can be evaluated under actual conditions. So these are the benefits that can be accrued. So obviously, we shall see what we did in this genomics assisted breeding components. When we started the work on this particular aspect, there is scanty information. Genomic resources were not available at all. We have to just start from the scratch. And we passed through our journey, passed through all aspects, developing different tools and till deploying these tools for product development. So I would like to spend some time on each one of these because it's a lesson learned for all of us. And I hope this could be of some use to our youngsters. So one of the issues, okay, those I have listed, I did not pronounce them. Mapping populations, identifying and validating marker system, new marker system. That's what we have also developed. Extensive phenotyping, genotyping, construction of linkage maps, QTL maps, then markers, validation, then employing them in M.V.C and M.V.C, developing backcross and introduction improved lines for future breeding, and of course, ultimately the product improved genotypes for cultivation. So we have step by step, we have gone through these, and I would like to narrate. It is not just my contribution. So especially, I would like to just remember a lot of people, when I go on, their names will crop up. They have contributed immensely in certain aspect, much more than me. But the spirit was there. So let's see what we have done. Mapping populations. Two important mapping populations were developed: TAG24, GPBD4, and TAG26, TG26 into GPBD4. So these were used extensively for mapping lately spot and rust. Mapping populations here, I mean recombinant inbred lines. At that point of time, the concept of recombinant inbred lines was very new. These mapping populations were some of the first mapping populations developed in groundnut across, we can say. Um, so What is the benefit of course, now we all know, and there are other tools also now at population level one can work on, but mapping populations are very critical in terms of how it compares with F2. The mapping populations, they are immortal. They can be multiplied, shared, and multi-season data can be obtained. You will come to know also. And more importantly, there is more chance for recombination in F2, F3, F4 early generation, so that the robustness of mapping will be much better as compared to F2 also there. So all these things really say that these mapping populations serve a very good purpose. So different mapping populations were developed mainly for foliar disease resistance, including introgression lines down below, using amphidiploid, artificial, this synthetic amphidiploids, that we will talk a little bit. And even parents or other traits were also used, and they could be really mapped also. One very important mapping population, which I would like to just highlight here, you find a lot of TME2 into TME2 narrow leaf mutant, many different traits, 430 lines developed by one of my students, Patanshati. Others names also will come anyway, obviously. This particular mutant was produced in IRI regional station at Hyderabad by work of M.V.R. Prasad and his colleagues from TME2 using EMS uh, or DES mutagen treatment. And they too differed for so many traits. So as a conventional mutation analysis, we can't really believe that such a thing could happen. That has happened in groundnut, and you'll come to know the reasons for it. And even several other crops it has happened. But in groundnut, and they differ for so many traits, and the mapping population gives much more variability, much more variation for several characters: morphological, taxonomic, productivity, drought-related traits, dormancy, iron efficiency, habit, seed size, so many other traits. I think there must be some untapped traits also yet, and many of them have been now mapped using this population. The other one, aflatoxin resistant TG series. The others, of course, are there. Anyway, I wanted to say a few words about this particular population, and so I said about that. Anyway, then of course, more important is phenotyping of. This is really crucial, especially current scenario. We have genomic tools, very cheap sequencing technologies are available. A lot of publications are coming through, many publications, much more than what we see in translation research, the products. So there is a big gap between what we publish and what we achieve. But in what we achieve, this particular aspect is really a big constraint currently for different rates. Phenotyping, this is very important. And we did uh, extremely good work in at the Harvard, especially because the conditions were ideal. Also, we created artificial epiphytotic conditions, raising plants in pots, infecting them with inoculum, keeping these pots in the field at particular point of time. That really made huge difference. You can see the difference in terms of incidence in different recombinant lines, how they are showing resistance to leaf spot and rust. Another very interesting thing about recombinant imbalance is, say, in different years, rust and leaf spot, their occurrence was different. And for example, in summer, rust comes up. So it is very easy to evaluate for both the diseases, same population, generate information on that. And over seasons we have done. And here in this graphs, you see some simple trend which can give insight into nature of resistance. As far as late leaf spot is concerned, it is almost it is normal curve. And for rust in both the cases, It's a typical one example I've shown at one particular point of time, one season, but this trend was there across seasons, varying with varying levels of occurrence of rust and late leaf spot, and two peaks as far as rust is concerned, indicating that there could be a major. In fact, our work led to that major QTL, one single major QTL was there for, whereas for leaf spot there were many QTLs. So this was indicative in a way. And of course, whenever we talk about genomics, we also talk about marker systems, and it has become very old-fashioned now to talk about markers. Everybody knows about them, but in groundnut, it was very scarce to begin with. There was a first report of RFLP way back in 1991 and 92. They found that there is no polymorphism in cultivated peanut or groundnut or as a species displayed considerable diversity. One of the first works was by Prakash Lab, E.N. Prakash. I happened to visit his lab, as it was told earlier, for 3 months, and that initiated me into this particular field of genomics related work in breeding. It is not hardcore genomics as such, it is foraying into genomics for breeding. So that's what happened. Of course, we all know that SSRs are quite widely used. We used also these, and there is a huge resource available at marker.kasusa. This, of course, we had important collaboration with them. DNA Research Institute at Kazusa. We'll come to know about it anyway. Then DART was used. And now, of course, the fashion is SNPs, because sequencing has become so cheap and it becomes so easy now to use them. And there is one more class, the last one, transposon-based markers. So this is a very interesting experience for us. So we will talk a little more about that development of transposon-based markers. So this talks about a lot of interesting things. One thing is, more we visit and talk to or see and visualize plants, more we learn about the biology. More we learn about biology, we'll come to know a lot about the plants and how to really manage what are the key insights we also get as one aspect. Other aspect is this work was curiosity driven. So that's very important aspect in our research, whether it applied or it is a very basic research. Curiosity should be there. That can lead to really good benefits. That's one thing. The other word which is often used in research investigations is, of course, all of a sudden we get a new thing, serendipity. They say. So this was something of that kind for us, serendipity, a new illumination for us. So let's see what is that. So here is one, of course, slide. In the transitional belt, as I said, two crops were in vogue, and Spanish bunch groundnut, especially early maturing groundnuts, were taken up. And sometimes they were came to harvest in September, October. By the time there was used to be some continuous rains, and crop suffered due to in situ germination. And interestingly, all Spanish bunch varieties, they lack this dormancy, what they call fresh seed dormancy, whereas our Virginia types do have this fresh seed dormancy. Even if they are caught up in rain, there won't be in situ germination. There won't be loss due to that effect. So one of our objectives earlier was to develop varieties, Spanish bunch varieties with fresh seed dormancy. So when very interesting material was there, which was there in the breeding bank when I entered, say, ACRIP. This was, we named it as Darwad Early Runner. publication is also there, small publication, international newsletter. It was a very freak, curious type. It had some very special features. It matured within about 90 days, very small pods, and sequential habit, but only 4 branches, each branch running almost 1 meter. Pods were very small. There was fresh seed dormancy in this line of 15 to 20 days. So this made us to treat this with mutagen and look for any erect types with large seeded with fresh seed dormancy that will be benefit for us. So when we treated with and grew M1 and M2, we just M1 pulled together and raised a very huge population. And myself and Nadaf, we were utterly surprised when we found the mutants. The frequency was so very high, and the types, all possible types. It was like a germplasm bank when we saw the M2 population. Though it suffered from some germination problem due to fresh seed dormancy, because we took seeds from summer crop to kharif crop, we never anticipated that there will be failure of some germination. Indeed, there was some effect on germination, but in spite of that, there were umpteen number of mutants of different kind. Just to have an example here, the ones looking at typical Spanish bunch, typical Valencias, typical Virginia bunch and runner types were seen, very high frequency. There were other features which were evident. And of course, Nadaf wanted to work on certain other aspects for a PhD work on iron absorption efficiency and seed dormancy, etc. But when we looked at this, really it motivated us to change his PhD program, and he worked very systematically on again treating and looking at systematically for each plant and its family, how it behaved, etc., and became with certain very interesting features. Very high frequency of mutations, reversions, you can see here. There were plants occasionally again in the mutated progeny, resembling our Darwin early runner, may not be in all features, but in certain key features. Non-random mutations, repetitive mutations. We'll come to know about disease resistance again. Or it could be whole family that we called homozygous mutations, all, each and every plant. There was no DER-like plant. Everything was mutant. How could this happen? Really, it was a surprise for us. Unstable variants. Some were, again, not stable at all. Somatic mutations were pretty common. Of course, as you have seen in this, multi-character mutations were pretty common. Or multiple alternate forms, like in the habit, uh, it could be erect, it could be decumbent, it could be recumbent, etc., etc. For other traits also, an array of multiple alternate forms for each trait. So at that particular point of time, we were wonderstruck. And of course, when we really searched the literature on mutations, etc., in plants, not much of this kind was there earlier. But in Drosophila, a lot of interesting work has been done, and it was shown that its activation of transposons could lead to some mutations of the carrying these features. So that we invoked transposon activation as a possible reason for this sort of a behavior. So this publication came way back in 1996 in Infitica. One of our other students, Sheshagiri, also did his PhD and MSc and PhD on this material. Nidaf did his PhD on this material. Then this publication came way back in 1996 in Euphytica. Even still, I remember about this publication. So we talk about transposons, jumping genes and transposable elements. Some few sentences about them. Of course, many of you know about this. There are two classes, retrotransposons and DNA transposons. The first one, copy and paste. The second one, cut and paste, because in the first group, RNA is generated from the transposable element and it gets converted back to DNA with invertase, transferase. Then class 2 these types, they are DNA elements. There are two again in this, autonomous and non-autonomous. Autonomous, the transposable element carry the key gene for transposase, which is responsible for jumping in and jumping out, present in the element itself. And the second class, non-autonomous elements, they lack this mechanism, of course. Much is known about it, but still it is a mystery. Non-autonomous elements, they multiply in great numbers, and how it happens, etc. In certain systems, it has been deciphered, and of course, in groundnut, it is not deciphered. Then in 2004, there was a publication for the first time in groundnut talking about transposon. I think here also Nadaf went to Texas A&M as part of this publication. His name is there in this. Patel et al. 2004, oleic acid mutant, in which they analyzed why oleic acid gets accumulated. What is the genetic basis of it? They found that the key enzyme, the gene responsible for producing key enzyme, it converts oleic acid to linoleic acid, has got mutation in both genomes, A and B genome. One point mutation. In the second one, there's a insertion of mite, a non-autonomous DNA element, mite. So is present, creating a frame shift mutation leading to this mutation. And obviously, these genes have been now exploited even in India. And Berasab also part of this, creating varieties which are resistant to high oleic acid or low high oil ratio types. Very profound advancement, and the starting point is here at real genomics level, genetics level. At that time, people are not talking about genomics; it's a molecular level or genetic. Genomics came later, anyway. At that point, it came, of course. Miniature inverted repeat transposable element, mite, it is called, and usually it is popularly termed as H mite, Rachis hypogia mite. In each species, there could be different families of mites. Here we talk about H mite 1. Of course, a typical mite has got some interesting features. It is interesting to know about this to understand a lot of other issues. Each mite species may have unique sequence, the internal sequence, terminal invert repeats of sizes, 10 base pairs and more. Here it could be 50 to 800, sometimes much more than this in some cases. And of course, there is another interesting point, that is flanking marker, 2 to 10 base pair. It is also called TSD. This defines the site at which it is incorporated, and even if it jumps out, we can make out where earlier this particular transposon exists, and therefore, very interesting feature, that is insertional polymorphism, which could be used for marker development, is associated with this. And high copy number, 3,000 to 10,000 per genome, is because they accumulate by amplification bus. Therefore, you'll come to know in mutants, it is not just one mutation. It is so many places this get incorporated within a cell. You'll come to know about it. Another interesting feature which adds to ideal feature for marker is terminal inverted repeat, 25 base pair approximately. Then another interesting non-coding interpersonal element is now genes. Of course, nowadays it's been shown that mites get transcribed into small RNA, and small RNA has got very important role. Now it is turning out to be RNA word from DNA word, and small RNAs have great role in genome organization, in regulation, and so many other issues, or in even expression of phenotypes, interaction with environment, etc. Interesting things are coming out, but as such, there are no genes which code for a protein here, like transposase, for example. Then another very interesting feature about mite is, unlike retrotransposons, these mite elements, even in other species, plants and animals, they also prefer gene locations or gene low copy number genes and their promoters, and therefore functionally they can be really very important. If they enter or come out, they can have real impact. Therefore, we see a lot of effect role in gene regulation, gene organization, species diversity, etc. Then one another interesting thing, of course, which we noticed first time in groundnut, genomic stress, mutagen, in vitro culture, or wide hybridization can lead to activation of these. This was also studied extensively in rice, and we learned a lot from rice work, but it ran parallelly. We never had molecular tools at the time as compared to rice, but we came to know that this is happening in groundnut, in our material, and what we saw in the field, we could really explain. So that's what. So at this particular point of time, Ramesh, but after spending 1 year in Australia, he went there as postdoc, know more about tilling and learn a lot of molecular tools. He applied thermal asymmetric interlaced PCR to isolate one of the sites, the flanking marker associated with H-mite 1. Of course, as I said, there could be several sites, but we wanted some sequence somewhere defined location to define the location. So he first isolated this, and of course, it was published in Current Science. And from TMA2, he called it as a festival linked site. And then we checked this. And one interesting thing is this is very easy because very large size presence absence, and agarose gel can be used just simply to study this at individual level. And we had, as I said, in DER, repetitive mutations. We had from DER one Valencia 1 line, which was resistant to rust, but susceptible to leaf spot. When we treated this with another mutagen, within a population of 200, we got 5 plants or 5 families which were resistant to leaf spot, which were sharing a lot of common features in pod and other features, varying only a little bit for some features like leaf size, etc. 4 families were there. And when we, of course, this was later developed into a variety. Of course, because of GPBD4, nothing to do with that. This did not come in for it. So what happened really is when we grew this material in subsequent generations, so we saw occasionally some plants which are turned out to be susceptible variants. So we also took this. And another key interesting feature about these mutants is some of these mutants were homozygous. Whole line M1, it was like VL1. M2, whole line was mutant, especially so with 28-2. Each and every plant was mutant. In that cases, couple of 1 or 2 plants or couple of plants. But in 28-2, it is all whole line. So how could this happen? Was the perplexing question for us also. So when these were analyzed for say this FST1, so it was interesting to find a association. It was absent in VL1, present in all lead spot resistant mutants, absent in all the variants which we got, which were susceptible to likely spot. So obviously, activity of this is associated with, uh, say, H mite. So this is quite at that particular site, FST1. So then we examined other interesting mutants we had with us. One is narrow leaf mutant TMA2, which I talked about earlier, which is Spanish bunch, narrow leaf mutant Virginia bunch. Yeah, mutagen produced. It was present in TMA2, absent there in this. GL24, we had isolated two spontaneous mutants, which are Virginia runner 1 and 2. They were absent there, present in GL24. TAG24, it is present. One of the somoclonal variant, PAJ24AM, it was absent. DER, Harvard early runner, it was absent there. In Spanish bunch mutants, it is present. Gamma reproduced Spanish bunch mutants. VL1, Valencia 1, a mutant from DER, again absent. But all these lines, which we talked about, and also SB7, Spanish bunch 7, it was present. So there is some sort of undercurrent of association of these mutations with activity of these transposons. Then we wanted to test across germplasm. We took mini-core from microsat. When we examined it, it was quite interesting to find strong association. It is not just presence absence anyway, but very high percentage of Spanish bunch and Valencia types belonging to subspecies Vestigiata. They had this, they lacked that. Some of them they lacked it. Subspecies Hypogia, majority of them they lacked this particular H might one, and only very few of them they had. So very strong association again. So these were published, of course, in some journals. Then at this particular point of time, I was happened to visit Brazil upon invitation for to attend International Peanut Symposium, where Kenta Shirasawa was also present. He an expert, excellent expert in genomics as well as bioinformatics. It's one of the his contributions are monumental in different crop species. The expertise is of highest order. So he had shown using the information earlier on mite from Patel's work as well as our work. So he looked at a whole genome of a few varieties, Japanese varieties, just 2 or 3 varieties, and using polymorphism, just 504 sites, 534 sites, 23% of polymorphism. So this was quite interesting. And of course, my visit there and interaction with him, of course, fructified into huge collaboration between Ramesh Bhatt and Kenta Shirasawa from Kazusa DNA Research Institute. Wonderful project, DPT for, and Indo-Japanese project, where they had occasion to visit, and he visited several times India. Students also went over there, and they looked at in some of our material the type of polymorphism what is there. So have a look at this. So it's quite phenomenal in all the varieties we used as mapping to develop mapping populations, including mutants. It's quite interesting to see in the mutants it's much more, 35.5% between VL1 and 110, came 26% to highest, again into specific crosses, higher level, obviously, and compared to even other varieties, there also there is significant amount. So Polymorphism is quite good. There, if genomics data is there, sequence data, it's quite easy to track them, even in the absence of it. So simple lab tools can be used to study. Could be mapping population, could be segregating material, or whatever it is, using site-specific markers. So using even simple tools like, as I said, agarose gel, they could be studied. Important tool for real application. So TE markers have become important tool for, especially Ramesh Bhatt has used it extensively in his group. These two publications are examples for it. Genome-wide structural mutations among the lines resulting from genetic instability in phenotype, and development of HTE markers through genome-wide analysis to develop markers, etc. In the next, it was used to map for association mapping, background selections, studying genome instability and rearrangements. You can have these slides also if someone is really interested. So this is the important output from curiosity driven research. You can very well imagine it can be really considered as monumental, comparable to the research contributions from other places internationally. So we are also capable of it, but we need to spend more time with the plants, try to understand, but also have knowledge about what's going on in other systems and try to apply it, try to collaborate. Collaborations is key again, and really we can come up with something innovative. So that's it, which is really useful also. That's it. So this is the key lesson learned. Come back to polio disease resistance, our main theme of this presentation, QTL mapping. So this is all started with good mapping population, good phenotypic data, and it is very easy to really get collaborations. At that time, of course, ever dynamic Rajiv Varshney, everybody knows about him and his achievements, his capacities to network, his capacity to access international genomic resources and funding and publications. So we started first collaboration. In fact, for him, it is one of the first collaborations in at ICRISAT. My student Kedikar was already spending time there. So when he started his work at Applied Genomics Lab, so he was principally responsible for, at that time, as I said, genomic resources are very scanty, access to internationally available genomic resources, very few though they were, and then provided the facilities, resources for conducting the genomics work, genomics work. Two of my students worked on initially, Sarvamangala and Kedikar. That was the publication. And then later, another student, Sujay, worked on increased number of markers on both the populations. In the first publication of that kind on foliar disease resistance on markers, the QTL, major QTL was discovered, and validation was done in across germplasm. 95% of germplasm, whether coming from interspecific derivatives or land races or varieties, they had this particular QTL marked by a particular, of course, markers. So that was a real important development. And then it was expanded later by Ramesh Bhatt, including age markers, umpteen number of more markers of different kinds. And by that time, there was, as I said, utility of real. We had 13 seasons data. That is quite phenomenal. You don't rarely find such data. So increasing number, they did very deep analysis. Publications also came through. But collating all that, 56, 45 markers, 188 and 181 in two different populations, 289 and 188 in two different populations. So there was more precision, more markers were added, and obviously many potential markers for applications were really unearthed. This was an important development. And again, highest point is collaboration, good phenotypic, the material generation, genomic, genetic resource production, phenotyping, which we really undertook at Dharwad. And naturally, collaborations come when we meet people with other capacities. Genomics work was done at ICAR-IIGR, and later, of course, quite extensively at biotechnology lab at US Dharwad itself. And there was also, obviously, collaboration with Japan. So all these collaborations are key themes, their developments. So whenever we talk about markers, another issue is validation, whether it works in practical situations or in different situations is one thing. Some interesting work has been done on all selected resistant lines were checked from recombinant impedance and backcrosses, and was found highly validating. And one another interesting, while developing recombinant impedance, uh, we need to follow, of course, from F2 onward single seed descent. And we know that in the groundnut, if we sow single seed, we may get the plant or not, we may get the pods or not. There is a lot of problems. So we used to sow whole line and pick up one good representative plant. Doing so in F3, for example, whole line, when we grew about 25, 30 plants, there were some distinct segregants in later generations, like in F3 and F4, which differed from the whole family. For, say, rust resistance or lately spot resistance, whole family could be uh, susceptible, or one plant could be resistant. Then they are called HIFs, heterogeneous inbred lines, if they are multiplied into a family, and they are almost approaching nils. Producing nils is very, very tough situation. So many backcrosses, etc. But these nils from HIFs, so while developing the genetic resources, so it is also possible to develop such very useful material. Yes, we need to, as breeders, really look at this also. HIFs were used and it was authenticated. Rust resistance was authenticated by one of our students of Meshpet, Srinivasappa Eri, other students that validated. Then we had interesting publications. At this juncture, sequencing has become a common thing. And of course, labs like uh, Applied Genomics Lab at ICRSAT, Evarat, along with, of course, Rajiv Vashne, Manish Pandey was there, and whole lot of other contributors from different institutes and international organizations were involved in what is known as QTL-SEC approach, selecting say extreme types, using them as a bulks, sequencing them, and taking the reference genome from diploid, which is present now in say Duranensis and Ipiensis, the progenitors of Brachyphycogia as reference. They fine-tuned the QTL, identified several sequences associated, and also developed diagnostic markers, three for Rust and one for LLS, which could be really studied even in small labs using agarose gel. So this is an important development. Then we had another very important development. Again, a strong collaboration between three institutes, but contribution of Kenta Shirasawa was monumental here. His capacities for data analysis, for genomics, and Ramesh Bhatt's leadership at US Darwad, and his contributions are really wonderful. And you find all earlier contributors in this reference list, along with Raju Varshney and Manish Pandey, who are part of this journey. Uh, then already some other sequence information was available from another material VG9514, which is again derivatives from same ICGV 86855, which is resistant to both rust and lead spot. This information was available, and tag 24 and GPPD4 sequence information was also provided by Varshney. Analysis, deep analysis, very deep analysis really led to identification of candidate regions and candidate genes. These were genes, foreign genes. So it has gone up to that level. So from the phenotype to the gene level in a very complicated diseases, quite a remarkable journey and involvement of so many. That's a beautiful part of it. Yeah. So then it's a long talk, sorry. Integration from synthetic amphidiploids. There was another journey. Nalini Mallikarjuna. She has done wonderful work at ICAR-IIGR. She is not there now at present. In generating interspecific amphidiploids using wild species, different accessions. We all know that groundnut is an amphidiploid, allopolyploid, and this event has occurred in nature long back, maybe once, and therefore it is really constrained. The genetic diversity constraint. Many earlier work used to tell about limited polymorphism, limited variability at G level, but when we look at the crop, so much variability, etc. This was a statement that used to come quite often in the meetings. So how to really generate new material, going back to the wild species, different species. So she had generated in this particular publications 21 different amphidiploids, and you can see the picture there, beautiful picture in a publication. And we picked up in a collaborative project funded by DBT between ICAR-IIGR, Genetic Resources Unit also, and Genomics Lab, and say yes, they were both biotechnology as well as genetics and plant breeding. We picked up two the specific hybrids, and then we undertook one Varsha Kumari, who did extremely good work on this in progression. There were some publications with the team. These are the two wild species or amphidiploid artificial. We synthesized amphidiploids using wild species, different accession. But this is one of our important bee contributors in both the species, and they were crossed with several of Spanish bunch varieties, as well as one ICGS 76 junior bunch. Back cross was attempted, and many resistant intervention lines were isolated. They were validated using all markers, both PhD, SNPs, material generated, and some of these lines were again treated with mutagen, and it produced very interesting lines with modern kernel features, as well as in addition to the LLS and rust resistance to early leaf spot missing material was added to this. Will be really very important genetic source for which our ongoing breeding projects. About introduction. Then lastly, what we all spent so much time discussing and researching and reporting, etc., was ultimately for molecular breeding. That's what we wanted to authenticate, deploying the resources for developing the varieties, products. That's it. So one of the attempts was made by Ramesh Bhatt again, uh, support from fund coming from different sources. So he attempted crosses with JL24, TME2, DH86, and ICG76. We all know that JL24 is really early maturing, really good for especially kernel size, very attractive. There is a great demand for these sites, kernels. That aspect we need to really keep in mind. Therefore, it's still popular in some areas. Other interesting point is partitioning ability, and also it has got some level of field tolerance to bud necrosis. TME2, of course, as plant breeders or groundnut workers in any field, we know all about TME2, 1940 released, but still so popular in southern India. So obviously for its ability to withstand drought, especially in say light soils or in frequent wherever frequent droughts occur, its management through source sink balancing. So it is able to put forth some really quick foliage whenever droughts there, where its remission is there, so it can come back with some foliage, and pods can be there, some pods and some yield can come there. Uh, so obviously pod bearing there, if you sow in these areas, high pod bearing varieties like GPBD4 or certain other varieties, whenever these droughts are there, their shelling percentage goes down because of competition between the source and sink. So this balance act is really very important, and we need to know as breeders get insight into agroecology and plant traits to come up with nice ideas to develop impactful products. Is very very important. Biology is really important, and of course the field performance and in gaining insight into why certain varieties like him very well is very important. Likewise, DH86 again, much shorter duration, and it has done very well in agri trails across zones, and even in Uttar Pradesh, Rajasthan, etc. And when we look at trait, important trait, it is high temperature tolerance that is there in this particular line, and also it has got tolerance to bud necrosis. Likewise, ICG 76, a VB good VB Virginia bunch cultivar, which is also sustains in mid-season drought, uh, and also it has got field tolerance, very high shelling percentage, good seed for kernel traits. So they were taken as recurrent parents, and foliar disease resistant material, and all of them GPPD4 and ICGB 80099, 900005, IL1 and IL. These are all from derivatives from wild species. So obviously that shows the importance of wild species generating genetic resource and genomic resource also. They were taken and crosses were effected. That cross was followed 3 to 4 generations. Then self-reproducing were evaluated. And two interesting lines, one derived from JL24 and another as a recipient parent, and another from DMA2. DBG3 and 4 were developed. Let's look at a little bit about these. Those publications have also come and look into it. The donor for DBG3 is ICGV 86669. Donor for DBG4 is GPBD4. When these lines were sequenced, to look at their credibility as essentially derived varieties, you can see that, of course, type of markers employed earlier for foreground selection, background selection, when they were evaluated, so many SNPs were evaluated, and you can see the donor contributions and mostly restricted to disease resistance. So that's the beautiful part of it. Essentially derived varieties, even at sequence level, you can see. So that's proof concept also. And coming to the performance, you can see across years resistance for LLS over seasons as compared to recurrent parents. Then reaction to disease, the diseases also like rust. It may not be perfect, but really good. 4-4 is really good. 5-4 really good. 6 is really good. 4 is excellent. This 4 is excellent. So this is the advantage as compared to 899 or 799 in these two varieties. It's a real important contribution in developing popular varieties, versions with this. Of course, that has now been extended to even oil quality also, along with resistance. But I will restrict myself to disease resistance. So these are the performance across different trials. 28% improvement, large-scale demonstration, farm trials, multi-location station trials, performance of M.V.C likewise 29%. Then ultimately at state level, they were introduced into ACRP, that is mandatory, but conditions of yield levels, etc., was taken into consideration. Voting, release, but at state level, because of their importance in growing areas and because of the area covered and because of the real appreciation by the farmers, they have been now identified and released for zone 3, 4, and 10. It is in testing is in progress, and in zone say in some zones it is been up. So this is it. So there was another interesting attempt also at ICRISAT much earlier to what Ramesh Bhatt did. This is of course restricted to West because at that particular point of time, uh, valid marker and QTL information was available for West when this was attempted, and only SSR markers were available. They were used, and process were made with the recurrent parent ICGB 9114, JL24, and TAG24. Donor parent JPPD4. Backcrosses were attempted. Markers were used. Foreground and background selection, and some very improved best lines were picked up. They were also sequenced to show that only those sequences specifying resistance came from the other regions they came from the recurrent parents. So that's it. So validated by marker resistance select. They are ready for utilization in any breeding programs. Even genetic resources. That's a satisfactory outcome, quite satisfying outcome. Okay. So, of course, I had mentioned so many collaborators, researchers, even other agencies, seed production agencies, etc. Apart from that, in research and development, these were the people who were deeply involved, very closely involved. Because we have to always remember the fund contributors from various sources, and it's not just my contribution. Ramesh is the also the team leader here, uh, and he has really taken forward a lot of things, continued until achievement is made. And then, of course, there were a lot of OPAs in different projects. You all know about Nadaf. He has already given a invited lecture. Then we had plant pathologist from US, Dharwad, Lingaraju, and Patil were associated project. In biotechnologist, Sport Nayak was also some projects. Then, of course, I have already mentioned about collaborators across institutes. Enda Shirasawa, stupendous. Rajiv, mostly incomparable. Upadhyaya was there. Nalini Mallikarjun, cytogenetic site, providing genetic resources. Earlier initiated earlier collaborations also and continue to collaborate. In all this development, Manish was a part of it. He is now become a key player in groundnut at ICAR-IIGR. Then we have a lot of students who have worked on groundnut, both with me and Ramesh Bhatt. They are the real important force, as I said earlier. Very easy to really impress them and very easy to get our ideas get translated into actual things. So they were there, their pillars. Of course, we should not always forget about real workers at ground level, field lab assistants. For several years, they worked with us and still they continue to associate with us. It's so heartwarming to remember all. So that's it. Thank you. And last, not the least, farmers, of course, they are the greatest inspiration for me and others. Here is one of the snaps taken way back in 2007, several farmers in a groundnut field. Thank you very much. So I enjoyed sharing my experience. You are free to really interact both today and anytime online. So thank you very much.
Thank you so much, sir. Now we will have some questions from the participants. Anybody?
Oh, sorry.
Hello.
Hello.
Yes,
I am
yes.
unable to. Yeah, it is fine.
Now session is open for discussion, but be brief and to the point. I will request all the participants if they have some queries, doubts, or other than this talk, they related to groundnut, they can raise here or in chat box also. So it is open. Dr. Manjunath, audible, no?
Yes, sir. Yes, it is audible.
So any participants want to raise any queries? Anybody having any queries? Okay. If not, then you can go ahead for vote of thanks, Dr. Manjunath.
That's how 8 minutes, sir. Just one.
Yeah, sir, I would like to take. Okay, yeah, please go ahead.
Gowda sir, namaskar. This is Praharaj from IIGR, Junagadh, sir.
Please.
Dr. Sir, thank you very much. You have shown the mammoth works, the potential, and then all the thing was really it is tough. It is very difficult to comprehend in one lecture. We may be needing 2 or 3 lectures. But
Oh, sorry for rushing through. Yeah.
But anyhow, it is a very interesting one. I think the younger people will definitely have benefited from your experience, vast experience rather, and you will see that it will be associated with us in time to come. Sir, my only prevention is we have a different constraints. We have a constraints for high yielding. We have a different, we say, late leaf spot and a rust also. Similarly, we need a lima chutney variety for farmer preference and a system adoption, cropping system adoptions. So
that's it.
How do you see that which we should first, then second and third? So which we should cater, which one should be taken care of first and second and then third? Because we are confused in all of things because everything cannot be done at simultaneously. And
Yeah, that's it.
So what is
complexity.
your personal feelings so that we can pinpoint it 1, 2, 3 points so that we can get over these words? Thank you, sir.
The most important issues which I raised also and stressed a lot of time is the agroecology and preference there and success of whatever varieties they are there. What are the drawbacks there in those varieties? Targeting those aspects and using all accessible germplasm, that is very important, and intensifying the work in that direction. We may be able to develop a variety for that particular situation. Since groundnut is grown in so much different varied conditions, that too in under rain-fed conditions, dryland situations, where each zone and within a zone there are so many different agroecologies, then it becomes very difficult to breed a variety which can perform across. Maybe under irrigated system we can, but under rain-fed condition, we need to really define those things. That is very, very important. And fix our target. Say multiple traits again. So I talked only about disease resistance, but go back to GPBD4, wherein I emphasized about pod and kernel features, the market demand for it, and then the location there, transitional belt, which we defined. Therefore, GPBD4, for example, if it is grown in other areas, very dry land, say in Anantapur or other areas, if conditions are congenial, it can come up and give good yield. But under majority of the years, where stress, intermittent droughts, etc. are there, shelling percentage, though it is 77, 78% in transition, goes down to 50 or 55. So that is a problem. So we need to really target and develop varieties of that. And of course, use technologies wherever it is feasible. Like, for example, disease research is now possible for a marker, or quality, it is now possible. So other traits may be possible or may be in the process of development, some traits. But as far as performance is concerned, productivity, it is outcome breeding. So that matters. Their evaluation is really very, very important. And generation of breeding material identification is really important. That art of breeding, the outcore breeding, should be there. And we may have publications, a lot of publications coming through for our genomics work. But you see the satisfaction and we derive by developing, say, varieties like GPBD4, and when it has got an impact over large farmers, large number of farmers, that's a great, and that should be the main objective, and it should be the major focal point and driving force, and other things will really catch up. The type of genomics work I have done, it is not just plant breeding work. It is the keeping or getting ready to apply molecular breeding to actual breeding. So, for example, if you want to develop a variety in particular area, we need to really work for that area, but use these tools to add those traits. So that's it. So outcore breeding is essential, and there is no substitute for that. We need to spend a lot of time, and we need to have a linkage, proper linkages with all stakeholders, and right germplasm should be accessed. So that is no substitute for that. That is very important.
Thank you, sir. Namaskar, sir. Thank you very much. Manjunath, please proceed.
Sir, Yanagi sir, you want to say something, sir? Yanagi sir?
No, Dr. Bhatt, please.
Sir, thank you very much for the wonderful talk, and you have been a true inspiration for all of us. And great work, great vision, and great work. Thank you, sir.
Thank you so much.
If there are no more questions from the participants, I would like to take this opportunity to thank Dr. M.V.C. Gowda sir for his wonderful and insightful lecture. It would be helpful for all the groundnut breeders in particular, and other researchers and other breeders in general for their future work. Thank you so much, sir. We were fortunate to listen to you, sir. Thank you so much once again.
Yeah, thank you very much.
And Dr. Manjunath sir can be requested to send the PowerPoint to nodal officer, and we can have a copy of that. It is very
Yes, sir.
pertinent. We can share.
Yeah, please share it with whomsoever it is.
Yes, sir.
It has been given,
We have
and
already
it
got
can be
the
shared with
PPT,
anybody. Yeah, please share with anybody.
We have already got the PPT, sir. We will share it.
It is excellent, sir. It is excellent in the contest and of course in the reference point of view. A lot of work is done in this. Thank you, sir. So nice of you. Thank you, sir. Namaskar, sir.
Thank you. Thank you very much.
So from IIGR on my personal behalf and from the institute and ICAR as a whole, sir, thank you very much for sparing such a at least 2 hours talk. It's not a joke, and that preparation also at this age. So that shows itself, that itself shows your interest to the crop and subject, how veteran you are in groundnut. And the mammoth work you have showed, that publication, contribution in silico, as well as field, as well as paper, publication, collaboration, that is a monumental task. Any youngster or junior breeder, those who are starting their career, how one can build up their career and what are the scope for them, so they can just have a glimpses from the work of Dr. Gowda. And we all, those who are currently in the system, maybe another 4 or 5 years are there, Dr. Bhatt, myself, Dr. Praharaj, or Tirupati and ICAR-IIGR, and we have learned a lot from this such type of worker. Sir, I pranam to you, salute to you. Thanks, sir. You are also as a member, you are also a honorable member in our REC. You have guided this institute for work and program. So I thank once again, and our youngster or our junior breeder, those who will be in helm of affairs in coming years. So you will be truly inspirational to them. Thank you, sir, from our side, and we will be interacting in future also. Thank you once again.